This is Group 4's Lab Report 4 on Metabolic Test Lab by This is Group 4's Lab Report 4 on Metabolic Test Lab by Vianka Sanchez and Lucas Perez. This metabolic test lab explores the metabolic capabilities of various bacteria using different media tests, such as phenol red carbohydrate broth, triple sugar iron, agar or TSIA, and oxidation fermentation medium tests. These tests help us identify what bacteria are able to ferment or oxidize and produce products like gas. The materials used for this lab are the following. So for medias, we used the oxidative fermentative media, glucose media, and we used 8 of these. Then we used the TSIA, we used 9 of these. For our carbohydrate broth tubes, we used 5 of the glucose broth ones, 4 of the lactose, and 4 of sucrose. For our cultures, we used A. fascicalis, P. aeruginosa, E. coli, C. frundi, S. typhimurium, S. flexneri, Staphylococcus aureus, and Proteus mirabilis. We also needed a loop and a flame to sterilize the loop. The materials used for this lab are the following. Methods. For the OF glucose medium, using a sterile needle, we took a single colony of the bacteria and stabbed the medium about 1/4 inch deep into the tube. We did this to 2 tubes, with the first one being labeled closed, in which we covered it with 1 ml of mineral oil, and we labeled the second tube open, where we put no oil and left it as is. We loosely kept both tubes to allow for gas exchange. For the triple sugar iron agar, we used a sterile needle. We touched the surface of the isolated colony and we stabbed the needle into the center of the loop, into the center of the TSIA medium, to the bottom of the tube, and we streaked the surface of the slant coming back up. Again, we left the cap loosely fitted. Lastly, for the carbohydrate broth tube, we used a sterile loop, and we took a colony of the bacteria and inoculated it into the corresponding broth tube, which was either glucose, lactose, or sucrose, and we also loosely capped the tubes. Methods for the So these are our OF glucose medium. They were green before any fermentation occurred, and we will discuss our results in the following slides, but just a little more info on the color change. So if the slant remains green, then that indicates a neutral pH and no fermentation occurred. But if the slant turns yellow, then that means there was acid production from glucose fermentation. And this means that the bacteria are capable of fermenting glucose, leading to a decrease in pH and a change in color. For our opened and closed tubes, so this is our results for the P. aeruginosa. In the open tube, there was a slight change in color, which indicates there was acid production in the presence of oxygen. And for our closed tube, it did not ferment glucose and therefore remained the green color, which indicates no acid production in the anaerobic conditions. So this our E. coli for the OF glucose medium. In our open tube, the medium did turn yellow, and due to the acid production under aerobic conditions. And for our closed tube, it also turned yellow, so this tells us that E. coli is a facultative anaerobe, so the medium also turned yellow in the absence of oxygen. Our E. These are our results for A. fascicalis. So A. fascicalis does not use glucose either oxidative or fermentatively, so the medium remained green in both opened and closed tube because it did not produce any acid. These are These are our results for our unknown 104 in the OF glucose medium. So the lighter change of color in the open tube and then no change in color in the closed tube can indicate that this bacteria is possibly non-fermentative and non-oxidative. These are the last results for our OF glucose medium is for our unknown 204. So based off these results, we can say that our unknown is a facultative anaerobe. Its change in color in both tubes tells us this bacteria can ferment glucose and produce acid with and without oxygen. The last results for our These are our results for the TSIA medium. So for A. fascicalis, both slant and butt remained red, which indicates that it does not ferment glucose, lactose, or sucrose. E. coli was there was also no change in color, suggesting that this bacteria also did not ferment either glucose, lactose, nor sucrose. And then for our C. frundi, the change of color to yellow indicates that it does ferment lactose and or sucrose, but also glucose. These are our results for the TS. Continuing with our TSIA results, we have S. typhimurium, so the change in color and thus but signifies acid production from glucose fermentation under anaerobic conditions. For S. flexneri, the red slant indicates S. flexneri does not ferment lactose or sucrose, and it also produced no gas. For S. aureus, the yellow change in color indicates that this bacteria ferments both lactose and or sucrose in addition to glucose. Continuing with our TS, P. mirabilis in the TSIA medium produced a yellow slant which indicates it fermented lactose and or sucrose, and the red butt indicates that the glucose fermentation did not occur and is minimal. For our unknowns 104 and 204, the yellow slant and yellow butt results tells us that both of these bacteria are able to ferment sucrose and glucose and or lactose. P. mirabilis
So for our glucose broth results, originally the glucose broth was kind of like an orange color, and now we can see that E. coli had shown growth that settled down on the bottom of the test tube, but that's usually just because of the weight of the bacteria. They can like be flying around. And glucose and S. aureus are in the same seed. They both have the settled down bacteria, but at the same time they both show fermentation of glucose by turning yellow, because originally it was kind of like a orange color. But on the other side, A. fascicalis kind of like stay on the same color, like kind of like a orange red color, which shows that it did not ferment the glucose very effectively and also didn't show that much growth. So for the glucose broth results on our unknowns 104 and 204, we can see that they both showed growth that settled down because of the weight of the bacteria, but it's usually flying around the test tube. And also, we can see that the media kind of turned like a little bit cloudy, and the switching color from orange to yellow also showed us that both of our unknowns they ferment glucose. So for the glucose broth results, so for the lactose broth results, we can see that the original color was a yellow, like kind of like a bright yellow, and E. coli showed growth in the test tube, but it also remained the same color, showing that E. coli fermented lactose. But on the other side, we have A. fascicalis, which showed the media that had no little or no growth, and it also turned kind of like an orange slash red, indicating that A. fascicalis is a non-lactose fermenter. So for the lactose, so in the lactose broth results for unknown 104 and unknown 204, we can see similar results to each other because on 104 the media showed growth that settled down on the bottom. Again, we could have shaken it was gonna like start swimming around, and it also showed that it's a lactose fermenter. It stayed the same color. I know on the glove looks kind of like an orange color, but that's the best picture I could find of it. But it originally looks yellow. And also unknown 204 showed that the media had some growth at the bottom also, but this one show cloudiness around the entire media. And the color also suggests that unknown 204 is a lactose fermenter. So in the lactose broth results for unknown, for the sucrose broth result, we can see that originally the test tubes were red, like a bright red, and now we can see that the media turned a kind of like a bright yellow on both sides, on the E. coli and on the A. fascicalis, and this shows that it did not only had growth, but it also indicates that they are sucrose fermenters. For the sucrose broth result, in this other side, on the sucrose bar results, we can see that the unknowns had a pretty similar results from the E. coli and the A. fascicalis, but here unknown 104 and unknown 204 both showed growth that settled down at the bottom of the test tube, again because of the weight of the bacteria, otherwise they would be like swimming around it. And also we can see that they change color from the bright red to the yellow, which shows that they're sucrose fermenters as well. In this other side,
Conclusions. For our OF glucose medium, P. aeruginosa is oxidative, meaning it will oxidize in glucose in the presence of oxygen, therefore there was only a color change in the open tube. E. coli is a facultative anaerobe and can ferment glucose both aerobically and anaerobically, leading to a color change in both open and closed tubes. A. fascicalis is an oxidative and fermentative negative organism, so it does not use glucose in either aerobic or anaerobic conditions, and therefore there was no color change in either tube. For our unknown 104, there was a slight change in color in the open tube, but not in the closed tube, and this could be due to minimal oxidative activity or alkaline end products rather than true acid production. Lastly, for our unknown 204, the results indicate that it is a facultative anaerobe because there was a change in color in both open and closed tubes. For our TSIA medium, S-typhurium is a glucose fermentator that does not use lactose or sucrose, which is why it had no change in color. Our S-flexneri results show it is a non-lactose and non-sucrose fermentator as well, but a glucose fermenter. A. fascicalis does not ferment glucose, lactose, nor sucrose because the whole slant remained red. For E. coli, it also did not ferment glucose, lactose, nor sucrose. Our S. frundi was able to ferment them, and glucose fermentation occurred, leading to acid production. Our P. mirabilis also was able to ferment lactose and or sucrose, but not glucose. And then our unknowns 104 and 204 both had a change in color to yellow, which tells us that they are both fermenters.
so for this set of conclusions, we're gonna have the three different broths that we worked on. We're gonna have glucose broth, lactose broth, and sucrose broth. And let's go by order, like how we had them laid down in the PowerPoint. So we're gonna start with the glucose broth. First of all, we have E. coli, S. aureus, and A. fascicalis. Those were the bacteria that we did, the known bacteria. E. coli shows significant growth. Also, this worked for all the bacteria that I said that they had growth, but they settled down in the test tube. That's just the settling of the bacteria. If we shake it, it's gonna go up. It's gonna go down. Like it doesn't. This doesn't demonstrate if they're anaerobes or aerobes. This is just for the growth. And we can also see like a little change in the color from orange to yellow because of E. coli being a glucose fermenter, as well as its areas that had some growth, also turning it cloudy, but also changing the color, showing that it does ferment glucose and produces acidic byproducts. A. fascicalis also showed growth, kind of like a orange red color with not that much growth on it, but this could indicate that it's a ineffective glucose fermenter, but it has an increase in pH. And for both of our known unknowns, we see growth, we see glucose fermentation, and this can be compared to like the E. coli and the fascicalis growth. It's pretty much around the same. Also, we have lactose broth. Lactose broth, on that one we didn't do S. aureus, where we have E. coli and A. fascicalis. E. coli show growth and broth remain yellow. This demonstrates that it is an effective lactose fermenter and acid generator. A. fascicalis is a non-lactose fermenter because it show almost no growth in it, and it kind of like did like a little change in color from or to an orange, kind of like red hue, indicating that it's a non-lactose fermenter. Unknown 104 show growth, also a it didn't change in color, and this show that is